Welcome to Nikolai's genetics lessons, and today I'm going to explain how viruses infect cells, how penetrate them, and I'm going to use example HIV-1 virus. And here is the steps, 6 steps, and here on the right I'm going to explain every step. So I'm going to explain step by step how this happens. First of all, viruses cannot reproduce on their own. Instead, they have to sneak into host cells, then use a mix of viral and host proteins to copy themselves and spread. Scientists study these proteins to learn how to stop viruses. For HIV, their work has led to the discovery of powerful medications that interfere with key steps of the virus life cycle, and for many people, what was once a fatal diagnosis is now a manageable condition. As you see, according to this picture on the left, first step is going to be attachment and entry. HIV infects a cell when proteins on its surface attach to the receptors on the host cell. This locks the virus and cell together, helping the virus get close enough that it can fuse with the cell. The interaction is specific. Viral surface proteins and cell receptors have to match. HIV has a protein that match receptors on a few cell types, but most commonly infects immune cells called T cells. The virus must attach to T cell receptor plus at least one co-receptor to start an infection. HIV medications keep the virus out of cells by messing up the match between protein shapes. No match, no entry. The drugs can act on cell or viral proteins. On the cell side, CCR5 antagonist change the shape of co-receptor, so HIV cannot fully attach. With post-attachment inhibitors, HIV binds its main receptor, but the receptor can't cause a shape shift needed by the viral protein to connect with its co-receptors. And on the virus side, fusion inhibitors keep the viral surface protein from taking on the right shape. Fusion inhibitors does not recognize its receptor on a host cell. Now let's move to the second stage. It's shown here as reverse transcription. So HIV releases its genetic information in the form of RNA into a host cell, and the RNA contains the information for every protein needed to make more viruses. A viral protein called reverse transcriptase copies the RNA into DNA. Aside from the fact that it contains HIV genes, the new molecule now looks like host cell DNA. So host cell mechanisms of protection cannot tell the difference if it is invader or if it belongs to the cell. So what we can do and what cell can do, it can inhibit reverse transcription. If reverse transcriptase cannot do its job, HIV cannot replicate. To build DNA, reverse transcriptase uses building blocks or nucleotides from the host cell. Nucleoside reverse transcriptase inhibitors look a lot like those natural building blocks, but with a twist: when incorporated, they stop the growing of DNA chain. On the other hand, non-nucleoside reverse transcriptase inhibitors act more directly. They bind reverse transcriptase and disrupt the enzyme's catalytic activity, which prevents it from building DNA. Now let's talk about third stage, which is integration. Integrase is a viral provided protein that works with the host cell proteins to stitch the viral DNA permanently into the host. It works because DNA is DNA with a host or viral. it all has the same structure. The integration of HIV genetic information into the cells means that once a person is infected, the virus is with them for life. Can we inhibit integrase? Integrase inhibitors are often the first medication used to manage HIV. Integrase inhibitors act on the enzyme's active site, and it can't integrate the viral DNA into the hosts. Instead, host cell machinery degrades the viral DNA. Now let's move to the fourth stage, which is replication. What is the replication? It is transcription and translation. Once the viral DNA is integrated, a cell uses transcription and translation to make proteins to build more viruses. Just like with cells' own proteins, RNA polymerase reads the DNA to build a messenger RNA molecule. A viral provided protein called TAT boosts the amount of messenger RNA made from the viral genes. Then ribosomes and cell provided amino acids build proteins. Can we inhibit replication? This is hard step to stop because it uses host cells proteins and processes that are essential for life. So far, drug developers have had more success targeting other parts of HIV life cycle. So let's move to the next stage, which is assembly and budding. At this stage, two types of newly produced viral proteins anchor into the cell membrane and help organize and gather everything needed to form a new virus. Next comes budding, where the new virus exits through the membrane. As it leaves, it takes part of the host cell membrane with it, now studded with viral proteins. This stolen layer also protects the virus and helps it infect additional cells or even a new host. So can we block viral budding? It is an interesting question. Although there aren't currently any drugs that specifically target the assembly and budding steps, researchers are studying potential inhibitors of proteins that carry out the final stage of budding and may prevent new viruses from being released by the infected cell. Once HIV virus has all these parts and leaves the cell, it undergoes a few last structural changes. Now it's ready to infect a new cell. So can we inhibit maturation? Mature HIV has a protective shell made of viral proteins called capsid that surrounds its genetic information. GAG and GAG-POL, this is the names of the surface proteins, need to be cut by protease enzyme into the active forms, and protease inhibitors prevent that cutting. So the proteins don't do their jobs during maturation. Capsid inhibitors work throughout the virus life cycle. During maturation, they prevent cutting of a key protein into smaller pieces that build the capsid. And as a result, without a proper capsid, the virus cannot form correctly and can't infect new cells. While right now there is no cure, effective HIV mitigations allow people to live long and healthy lives. Medications can also greatly reduce the risk of spreading the virus to others. And if HIV is left untreated, it can lead to disease that badly damages the immune system called AIDS or acquired immunodeficiency syndrome, when even minor infections that healthy person's body can easily manage can be very dangerous or fatal to a person living with AIDS. And this is all for today. Subscribe and see you in the next video. Goodbye.